Vaccines. So, in this session, we are going to look at just the basic concept of vaccines and understand passive and active immunization, and that they are the principle behind developing all the different vaccines that are available today in the world. So, let us look at the learning outcomes of this session. Protection and prevention can be both due to passive immunization and active immunization. Passive immunization is raising antibodies in another animal and then using those antibodies, injecting or giving it to a subject only when essential. Active immunization is a process by which one's own system develops immunity against a pathogen. So, designing for active immunization is very important. There are several types of vaccines like whole killed organisms, attenuated vaccines, subunit vaccines, recombinant vaccines, etc. Now, let us understand how the study of vaccines began. So, it was Edward Jenner in the 1790s who discovered the concept of vaccine and also the first vaccine itself. So, Edward Jenner was working towards immunization against smallpox, and this was in fact his discovery and contribution to the world of vaccines. What he was when he was studying the smallpox, disease, and was trying to look at how this can be reduced, because what was observed is that smallpox was actually leading to a lot of fatal conditions, and that was something that was quite worrying, and therefore he wanted to bring out something that would be able to prevent the deaths. Now, he suspected, as he was studying smallpox, a connection between milkmaids who used to constantly tend to cows and they getting cowpox. So they used to get cowpox, but they did not suffer from smallpox. So he felt there is some connection between the cowpox and the smallpox, and probably that the milkmaids getting exposed to cowpox were immunized against the smallpox. That was his thinking, that was his understanding. And so, what he did is he took a scab from a cowpox lesion and he put the scab into a cut that he made in the arm of a young man. So the cut that he made was purposeful, and there he basically rubbed the scab. So he allowed the person to actually get the symptoms of cowpox. And the symptoms of cowpox are in relation to or relative to smallpox were quite mild. So the young boy, then, having been, inoculated with the cowpox, was then injected with smallpox. And as he kind of thought that the cowpox somehow immunizes a person against smallpox, that hunch came true when the boy had symptoms like the cowmaids, but did not have the deleterious effects of smallpox. And so, this was conclusive of the fact that cowpox is able to, when infected with cowpox, then the person gets immunized against smallpox. So, in other words, the person is able to develop an immunity or develop an immune response that allows it to also protect from smallpox. Now, the word VASA stands for cow, and that is how the vaccine, the origin of the name for vaccine was, because it basically looked at the cowpox and therefore vaxa for cow, and that is how the word vaccine originated. So Edward Jenner is definitely someone who brought this concept that when you inoculate a antigen within a body, the body will produce an immune response that can protect a person from a disease. Then, of course, you have Louis Pasteur, who has contributed a lot in the field of vaccines, and he was responsible for inventing the second generation vaccines. So, Louis Pasteur and his team were actually experimenting with a lot of pathogens. They grew this in the lab, cultured them, and then injected these cultures, pathogenic cultures, into animals to basically learn more about the disease and find out how they can be handled. Now, in one of the experiments where they were trying to inoculate cholera, that is Vibrio cholera, into chicken, um, you know, what happened is that one of his colleagues, after inoculating the culture in a media, uh, grew it in the media, but left it in the media for some extended period of time, in the sense that instead of taking the culture and inoculating in a stipulated period of time, he left the lab for some other work, and the culture just was kept in the culture media, uh, and was not used for inoculation. So when he came back, he took the culture and then injected that culture into the chicken. And what they found is that the chicken did get certain symptoms, but it did not have, or there were the cholera was not, or the manifestations of cholera was not observed in the chicken. So this was a very interesting observation, and one could say this is serendipity, because they just without really realizing what will happen if one keeps the culture for an extended period of time, if one incubates the culture for an extended period of time, what is going to happen? This was not something that they had thought at all. But on coming back and using that culture to inoculate the chicken, they found that the chicken would not get manifestations of cholera disease. So the chicken when re-inoculated with another culture, so this time they inoculated with a fresh culture. These chicken who got these cultures were not suffering from cholera, which means that the chicken had developed immunity against the cholera because of this culture. Now, therefore, what Pasteur concluded was that that because the Vibrio cholera were left in the petri plates for a longer period of time, and that meant that there is a stress level that the Vibrio cholera kind of went through, and in the process, the Vibrio cholera may have become weakened or attenuated, so their virulence becomes less, and therefore when injected the antigenicity was there, but it wasn't able to give rise to, or the virulence had decreased. So therefore such cultures rendered immunity, and therefore attenuated cultures are what are the second generation vaccines. So with attenuated vaccines, he tried it out for several diseases, and it was successful in getting anthrax, rabies vaccines, other than, of course, the cholera vaccine itself. Now, let us look at the basic concept of vaccines. So, the main idea behind developing vaccines is how do you protect people, or how do you protect animals from certain diseases. So, what is important is that if one needs to protect from a particular disease, then it has to strengthen and prepare the immune system to fight that particular pathogen, to clear out that particular pathogen from the system. So, this process, or this quality is basically due to the principle of what is called as artificial active immunity. Let us look at what we mean by this in some time. So, immunity can actually be classified into passive immunity and active immunity. Now, passive immunity itself can be natural passive immunity, or it can be artificial passive immunity. So, what is natural passive immunity? When an infant drinks the mother's milk has antibodies to fight several diseases like typhoid, etc. So, then the infant is getting immunized thanks to the antibodies that are present in the mother. So, infant is not developing an the antibodies. It is not producing the antibodies, but it is getting the antibodies from his mother. So therefore, this is natural passive immunity. Now, what is artificial passive immunity? So, for example, suppose a person is bit by a snake, then an anti-venom as an antidote can be given to the person and the person can survive. Now, this anti-venom has been developed in another organism. Okay, so what can be probably added to the person is a horse anti-venom. That means the antibodies against the venom have been raised in the horse, and those antibodies are collected and then injected into a person when the person is bit by a snake, and when the person has to be given an antibody to clear the venom from the body. So that is artificial passive immunity, which means the person is not developing the antibodies, but he gets the antibodies from outside. Similarly, active immunity is natural active immunity and artificial active immunity. So natural active immunity, say when say a person suffers from a particular disease. Say for example, in the childhood one gets chicken pox or suffers from chicken pox, or say a person suffers from chikungunya. Then uh, once he is exposed to the chikungunya virus, then the person can develop an immune response and the body can remember the immune response that it has developed, so that the person is protected from chikungunya for a long time. So that is natural active immunity. Artificial active immunity is when a person is purposely inoculated with a pathogen, but of course the pathogen's virulence is very less, or there is no virulence associated with the pathogen. So something has to be done to the pathogen in order to ensure that the person is exposed to the antigen elicits an immune response, but the antigen does not cause the virulence. So just like we saw in the case of the cholera, Vibrio cholera, when it was weakened or attenuated, it was able to raise an immune response in the chicken, and the chicken got immunized against the actual virulent pathogen. So that is artificial active immunity, and this is where the vaccines are basically coming in. So all vaccines, or most vaccines, are based on the concept of artificial active immunity. Now, successful vaccines have two attributes. One, it has to generate a strong immune response, and second, it should be able to give lifelong immunity. And a strong immune response definitely depends on the T cell activation and the B cell activation. And in fact, even lifelong immunity depends on these two per se. So when B cells are activated and differentiate into plasma cells, they give rise to antibodies, and they also differentiate into memory cells. This is important. And immunologic memory is very important for a vaccine to be lifelong, to give lifelong immunity. So these plasma cells would give rise to antibodies. So what you are raising is a humoral immunity. And when T lymphocytes are activated and differentiated to give rise to effector T lymphocytes, then these will function as cytotoxic T lymphocytes. So this is going to develop a cell-mediated immunity, and this is going to develop a humoral mediated immunity, and both are able to contribute towards a strong immune response and a lifelong immunity. So let us look at passive immunization. So conditions like pathological conditions in people would entail the need of passive immunization. Say, for example, some people are born with defects in the immune system and have immunodeficiencies, then they would need to be given antibodies from outside to fight the diseases. Or when a person is suffering from multiple disorders, so the body may be able to produce an immune response to one or two of the infections or the this thing, but may not be able to do so for other infections. So then one needs to be given antibodies from outside, or there has not been an proper immunization regime, and because of which the person may suffer from the disease and then instantly would have to be given uh, antibodies from outside. Now, passive immunization has certain limitations. But what is advantageous about passive immunization is that it can give immediate protection. But the limitations is that it may not give lifelong immunity. Why? Because the person's body is not developing the immune response. It is in fact getting the immune strength from an outside agency. So exogenously, the antibodies are given. Now, because the antibodies are raised in another organism, it is possible that there is a certain level of foreignness associated with those antibodies, and hence the antibodies themselves could act as antigens for the person, and that person could suffer from what is called as certain hypersensitive reactions, basically to in the process of removing the antigens that have been the antibodies that have been injected. So what we are looking at is that passive immunization has this limitation that possibly the antibody that has been injected itself can elicit an immune response. So these are certain cases where passive immunization is needed. For example, botulism, diphtheria, hepatitis, measles, rabies. So when you have a dog bite, immediately you need to go and get an injection, which is basically anti-rabies injection. So these are certain diseases that are certain conditions which need passive immunization. Now, active immunization, many of these vaccines that have developed based on active immunization are able to give life-term protection. And there is a national immunization scheme which is given to people to protect them from several diseases. So there is a list of vaccines, and in fact, a very well planned schedule is already available for when the first dose has to be given, when the booster dose has to be given, and so on and so forth. So the protection is basically from hepatitis B, or the vaccines are for hepatitis B, for DPT, that is diphtheria, pertussis toxin, and tetanus, OPV or IPV, that is the poliovirus. So OPV stands for oral poliovirus and inactive poliovirus. MMR, that is measles, mumps, and rubella. Haemophilus influenza, varicella zoster, which is specifically for chickenpox, and pneumococcal conjugate vaccine. Now, all of these are given to from the infants to the teenage age. And in adulthood, also, a lot of vaccines are given specifically for certain viruses, as well as, say, for example, hepatitis A, influenza, and this may be needed to be given quite frequently, depending also on the fact that whether there is a pandemic that has come up or not, or whether there is an epidemic that has come up or not, that way. Now, what has been observed is because of these national immunization schemes, over a time, the number of reported cases for these diseases have decreased. Where once polio used to be a very common phenomenon, now you will hardly find it in the population. So that is because of continuous or constant monitoring of whether the children have been vaccinated or not, that they are protected from many of these detrimental diseases. Like, for example, in this case, BCG was not mentioned, but BCG is also given. BCG is for tuberculosis. Now, designing vaccines. So, basic research is always very important while designing vaccines, because whatever vaccine is being developed, that has to activate both humoral and cell mediated immunity, and not just that, it has to develop an immunologic memory. So incubation period of the pathogen is very important. For how long a person is exposed to that particular inactivated pathogen or weakened or attenuated pathogen is very important. How many doses has to be given, and what is the dose itself is very important. And of course, the route of administration, whether it is to be given intradermally, whether it is to given orally, whether it is given, it is to be given through the nasal this thing, or just as a intramuscular injection. All this matters. Now, this, say for example, the Salk vaccine for polio, what has been observed is then vaccine is given. In the first 2 to 3 weeks, you will see a very heightened production of serum antibody, and then over months, the production of serum antibody may decrease, but what is definitely happening is that the immunologic memory is increasing. And therefore, this is what is important. Because the immunologic memory is increasing, the persons are protected from polio lifetime. So that is important to understand. So while developing the vaccine, one has to ensure that it is able to increase immunologic memory. So let us make the conclusions. Vaccines are based on the concept of passive and active immunization, as both are able to neutralize the pathogen or causative agent. Passive immunization can be natural or artificial. They give immediate protection, however, not lifelong protection. The antibodies are raised in other animals. Active immunization is when one's own body elicits an immune response, either humoral or cell mediated, with a thrust being on developing an immunologic memory. The design of vaccines will have to ensure a longer exposure of the antigen to the body so that the body can develop a high immunologic memory, and therefore that protection is going to be lifelong. So when bit by a snake, an antidote for venom can save the person. When one suffers from chickenpox, one may get immunized against it lifelong. These two aspects are the key to fighting several pathogens, because of which you have vaccines being developed. Thank you.